just means we'll have to pick somewhere when we get back. Yeah. Morning. Good, how are you?
Could get you in a couple of them. There you go. Great. done it. Yeah. All good. Yep. Okay. Yep. No stress. Cool. Right. Bye. there.
Can I just make a few corrections to my presentation? Is it possible to upload it,
Yes.
the new version?
Sure.
Actually,
This is your, yeah. Okay. Just thinking about it, the best way to do it would be to hyperlink to in so you're the expert, I do whatever you. Okay, cool. Just, yeah.
Yeah, meetings.
There you go. Good. Happy last day.
I know. Yay!
You're okay for everything this morning as well?
Yes, it's all
Okay,
good.
perfect. And timing?
Same
for
as
morning tea 10.
Yeah.
We got lunch at 12 and then afternoon at 3.
yeah, that's is it 10:30, isn't it, morning tea?
I got it here at 10 o'clock.
No, that's not us. 10:15.
So 10:30? That's all right, we can take it now.
No, but it's not. I was like panicking. I'm like, I don't have a USB. I have to take the whole computer down.
No.
10:30 to 11.
No, I need
Yeah.
It's like the same as
Oh,
every
so-
other day.
okay, yeah.
Exactly the same as
No
every
worries.
other day.
Yeah, okay.
So
So, yeah, 10:30 and then.
I don't think so. No, that's all wrong. They're all completely wrong.
We just go back to yesterday.
Yes, it's supposed to
be every same year.
That's all right. I'll put in the no draft at all.
Yeah.
Like,
What's clear on the it's exactly the same as
yeah.
so it should be like that.
Yeah.
That's more than just
Yeah. No, yeah. It's probably just my system that's called it wrong because I got the water at 10:30 as well. But when that section put on Wednesday as well, right?
Yeah, that's right.
Yeah, so like the normal ones. Yeah, that's all right. I'll go change all that.
And I'm pretty sure that that's not, like this is 3 till 3:30.
Yeah. Yeah, no, sure. I'll just change that. In my computer system, that's putting it all in.
I probably just didn't.
No, that's all right. Oh, sure.
Not that everyone's gonna be here. She's becoming it. But I keep breathing.
So we'll just I'll go and it's really long, So what I can do, so we're just making sure that the responsibility exercise. Yeah, it's a special practice. That's what they specifically shows. So the credits, we do all of our words and that I'm fine because I copied
There is some differentiates.
Your mic is again.
All right. Good morning, everyone. It's great to see so many, fairly rested faces in here. Thank you very much for coming. Before we start, I would like to acknowledge the traditional owners and custodians of the land on which we meet today, the Boonwurrung people of the Kulin Nation, and pay my respect to their elders past and present, and the elders from other communities who may be here today. And of course, a conference without the sponsors is not a conference, and it is really highly appreciated by everyone in this room. So I would like to acknowledge Verbeck, Elsevier with our journals, IGP, DDR, and POR, WeHi, and New England Biolab. So, as you all know, this session is dedicated to the Concepts in Parasitology course, and I just would like to spend 30 seconds on the idea of the course. So the idea of running a course was around ever since I joined the society, and probably 30 years before I joined the society. I found a paper from Graham Mitchell from 1981 where he talked about the prospect of putting something like this in place. And then in around 2010, 2012, those plans actually precipitated. And I think this is really driven by the fact that a society like the ASP faces challenges. And the main challenge is probably the increasing discipline fragmentation. So we all define ourselves as parasitologists, but of course, we are also molecular biologists, people working in genetics, vets, medicos, biologists, and we all have our particular view of the world. I think this is actually a strength of the society rather than a weakness, but this is something that needs to be harnessed. And therefore, we thought within a course like SIP, we would like to give people the opportunity to explore the full breadth of parasitology in a 2-week course. You will see where people actually going in those 4 presentations we have today, but I would like to acknowledge here the vision of so many of the society, and apologies to the people I don't mention by name, but I would like to say thanks to the executive under the stewardship of Dennis Doolan, Robin Gasser, David Emery, and now with Una Ryan, because without their support and vision and willingness to make it work, this course would never have seen the light. And of course, nothing happens in the society without Lisa and Nick. So also big thanks to Lisa and Nick. The course is completely run by the society, independently financed, and it's subsidized by your contributions, by your membership fees. But the biggest contribution, and this is, I don't want to play that down, but the biggest contribution is actually the time the lecturers put into this course. I made a back of the envelope calculation, and at some stage I arrived at $500,000 for a 2-week course if we would want to run it commercially and everyone would charge the hours they put in and the time and the equipment they provide. So it's a real fascinating story. So today we wanted to highlight the future of parasitology, because I have the enormous privilege to observe the future leaders in parasitology for 2 weeks being at the course. And we talked about in this conference that it's hard to maintain the knowledge in the field, that universities actually scrap the jobs and don't replace emeritus positions with retired people with new parasitologists. But I'm actually quite confident seeing the enthusiasm, skill, and talent every year about the future. So I think parasitology will go great places. And finally, I would like to thank everyone for entrusting me with the trust of putting something like this course together. That's a really great show of trust and confidence in me. So I'm really humbled by that. Thank you very much. Without further ado, I would like to introduce Dom Marendi. So Dominique is a senior scientist at the parasitology lab at Elanco Animal Health. And as you can see, Dom is always busy. She was always busy at the course. And because obviously being a senior scientist in a busy lab is not enough, she's also doing a part-time PhD at the Uni New England. I asked everyone, how did you end up there? So she said she did an honors project on wool quality and worm resistant using food supplement. And that eased her into she caught the bug there. And her favorite parasite is Fasciola hepatica. So outside of the lab, she tries to explore new places and going to restaurants with her friends. Please come up to the stage, Don.
Thank you, Alex, for that introduction. Oops. So today I'm going to be talking to you about the first trial of my PhD, which looked at risk mapping of liver fluke using a combination of plant and soil technologies and statistics. So globally, Fasciola hepatica costs livestock production industries an estimated $3.2 billion per annum. Nationally, sheep and cattle producers spend an estimated $10 million per year on flukicides, with additional costs in different areas of production. So between the year 2000 and 2015, it was reported that between 5 and 52% of sheep and 26 to 81% of cattle were infected with liver fluke. Over the years, while producers, many producers will know their infection status, like with a leader's presentation, a lot of producers will say, oh, I don't have fluke on my property, so why do I need to test? And processors are now putting in these electronic data capture systems and looking at how they can feed back the information from the carcasses that they're getting through their systems to producers to give them information about how their animals are performing and also about disease statuses. So producers are really going to have no option but to know their disease status on their property. So the life cycle of liver fluke is very well documented and understood, and it involves both a definitive and intermediate host. The parts of the life cycle that my project really focused on were the intermediate host and the free living life stages. These life stages have specific ecological requirements which geographically restrict this zoonotic disease. So the intermediate host are snails of the Limnaeid family. In Australia, the primary intermediate host is Limnaea tomentosa. In the 1970s, other species of intermediate hosts were introduced that are also capable of hosting these intermediate or these larval life stages, and these include Limnaea columella and Limnaea viridis. And there's a little bit of conjecture about these two intermediate hosts, with the potential that they could spread this disease more tropically because they're better adapted to tropical climates. So the ecology and distribution of these intermediate hosts is quite well documented, as you can see from the map on the screen. And the features of the ecology of Limnaea tomentosa have been really well defined by researchers such as the late Joe Boré. One of the difficult things about establishing correlations between the intermediate host and infection risk for livestock is that it's really difficult to establish a correlation between the presence of the intermediate hosts, the density of the intermediate hosts, and infection rates in both the intermediate and the definitive hosts. And this is because Limnaea tomentosa has quite temporary habitats. They're really good at establishing populations quite quickly, but they can also be flushed out of the system quite quickly. And as well, even if you find a population of snails that's quite densely populated, infection rates can be quite low, at about 1 to 6%. So it's important not to just look at mapping the intermediate host, but where exactly are we finding Metasaccharida in the environment? So that's the infective stage. So with the free living life stage, environment obviously affects their distribution as well, and their ability to develop and sustain their viability. So I just wanted to talk you through some of the very cool pictures that I have up on the screen. So the first picture that I have on the far side of the screen is the Fasciola hepatica egg. And then this very cool video in the center is a video of Sicaria shedding from a crushed snail that was artificially infected in the lab. So the important thing to understand about Fasciola hepatica is that one miracidium infecting a snail can self-replicate through 2 larval stages and produce up to 4,000 Sicaria into the environment. And then finally, we have the encysted metasicaria stage. So within this intermediate host, as I mentioned, we have the 2 larval stages that can undergo self-replication. The radial stage of Fasciola hepatica. So the first stage that released from the sporocyst, they're called the mother radii, and then every generation of self-replication after that is called daughter radii. So the successive nature of this, these generations doesn't change from environmental factors, but what does change is the number of radii produced at each generation, and therefore the number of radii that shed from the snail eventually. And in addition to this, temperature and being maintained in a moist environment is going to affect the viability of these metasicaria once they last in the environment. So there's been many forecasting systems developed over the years, and earlier models just looked at things like temperature, rainfall, and evaporation to predict seasonal risk patterns for Fasciola hepatica. But the limiting factor for these is that they're really geographically isolated and can only be applied to the areas in which they're developed. So you can't take one from Ireland and try and use it to predict risks in Australia. So more recently, GIS mapping, which involves computer-based analysis of multiple layers of GPS-linked data, is being used. And this has been done to map risk of Fasciola hepatica at a national scale in many countries, but has not yet been done here in Australia. So in agriculture, we also have the development of on-the-go plant and soil sensing technologies. And what this might allow us to do is map the risk of parasite infections at a more localized scale. And what I'm talking about here is mapping at an on-farm scale to help inform farmers with their integrated pest management strategies. So drawing all this together, risk of infection is determined by the presence of the intermediate host, which will therefore determine where metasicaria occurs in the environment. So by utilizing our knowledge of snail ecology, GIS mapping could help identify infective risk areas on farm. Additionally, this could add new measurements such as soil conductivity, optical reflectance of the pasture, and elevation to our understanding of snail ecology. So to validate this concept, we set out to survey pastures for both the intermediate host as well as the infective life stages of the parasite, and then correlate this with environmental factors such as soil moisture to identify high-risk areas on farm. So the important thing to note about the technologies that I'm going to talk to you about today is that this technology is already being developed and in many cases already implemented by producers to monitor their on-farm productivity and performance. So a good example of this is optical reflectance of the pasture. So this measurement is really easily accessible. There's a website called Pastures from Space, and it's also being developed to be hooked to drones. And producers can use this to calculate the amount of feed that they have available in a paddock to inform their grazing management systems. And so what the aim of this study is really trying to do is not create a new technologies to farmers for farmers to use and invest in, but look at what technologies they're already investing in and give them opportunities to utilize this to better understand parasites and their risk and inform their integrated pest management strategies. And so began the first trial of my PhD. So the aim was to assess the feasibility for use of GIS mapping technology to map infective risk areas on farm. The property that we used was located in the New England region of New South Wales and is a combined sheep and cattle property. And this property had a known history of liver fluke. Sampling was done in February of this year based on the reported seasonal prevalence of Limnaea tomentosa in the area. So I'm just going to quickly try and talk you through a bit of the paddock and some of the features that you might not be able to see from this image. So the elevation in this paddock varied by 45 metres. So the highest point of this paddock was along the most westerly fence line. It sloped down towards the creek before rising again towards the dam and the northern fence line. It then sloped down towards the lowest point of the paddock, which is just here in this most southeasterly corner. So to map the paddock, and we attached 3 pieces of equipment to an all-terrain vehicle or an ATV. The first piece of equipment that we utilised was a digital GPS, just indicated under the green arrow there. So this piece of technology not only recorded our elevation, but it marked the point geographically at which each measurement was recorded. We then utilised what we nicknamed the EM38, which is an electromagnetic induction sensor to measure our soil conductivity. So this was towed along behind the ATV, 2 metres behind the ATV on a rubber sled of approximately 10 millimetres thick. The next piece of technology that we used, we referred to as the crop circle. So this was measuring our optical reflections of the pasture, looking at the relationship between near infrared and red light as a normalized difference vegetation index. So once we'd connected these to the ATV, we took our ATV for a drive through the paddock. So this image here shows you the path that we drove through the paddock. And so there's a couple of things that I want you to note about this path. The first is that in both the top corners of the paddock, you'll notice a very tiny little orange dot, which is outlined in black. So while this looks like a line, it's actually thousands of these little dots, which is each point that a measurement was taken while we drove. So it's thousands of dots laid up upon each other, overlapping. And the other thing that I want you to notice is that we weren't able to drive through the creek, or at least we didn't attempt it again after we got bogged. And we also didn't drive through the dam because we didn't want to go swimming. So once we'd collected all this data from our drive, we went back to the lab. We downloaded our 3 measurements, so our NDVI, our soil conductivity, and our elevation, and we individually normalized each of those measurements. We then statistically analysed them with a kriging software called Vespa, which put them into clusters, which we were going to predict as our high-risk cluster, our low-risk cluster, and our medium-risk cluster. We then pulled this into our GIS mapping software called ArcGIS, which took these individual points and turned them into contiguous surfaces, which looked a little like this. So these are our clusters, and to explain these clusters a little bit more, we have our high-risk cluster, which had our lowest elevation, but our highest soil conductivity and our highest NDVI. We had our medium risk cluster, which had our medium elevation, medium soil conductivity, and medium NDVI. And then we had our low risk cluster in the orange, which had our highest elevation, but our lowest soil conductivity and our lowest NDVI. So once we'd created these clusters, we randomly selected 10 sampling locations within each cluster. So this is them here, and then labeled site ID 31 to 60. We then, based on the literature that we know about Limnaea tomentosa ecology, also selected 6 additional sites, just circled for you in red. So these 6 sites fell either, so 4 of them were directly in the creek line. So I had to get my gumboots on and go for a bit of a swim to get some samples there. And then we also had 2 samples that were sample sites directly adjacent to the creek line. So at each sample site, which we navigated to using our digital GPS, we took several samples. The first sample was a pasture sample, which we put in a brown paper bag labelled by sample ID. Took a soil sample, put into a labelled jar. We also spent half an hour at each site looking for the intermediate host of snails, and we collected any snails that we found, including any empty shells. And then at each sample site, we also took spot soil conductivity and NDVI measurements to make sure that we had correlation between what was being recorded at each sample site and what we'd recorded in our initial drive through the paddock. So once we got back to the lab, we examined each of the pasture samples under a dissecting microscope, and I sat there and counted every Metasicaria on the pasture, and then for all the samples, and then we dried each of the samples out to get our weight of kilograms of dry matter. For the pH samples, they were soaked in deionized water for 5 minutes before taking a pH reading using a pH probe. We examined all the snails that we found under a dissecting microscope and identified them with the use of a dichotomous key before crushing them to assess their infection status. And once we collected all these data, we statistically analysed them using linear regression analysis. So could this potentially work? So unfortunately, due to time and labour limitations, we were only able to sample 3 of our low risk, 5 of our medium risk, and 10 of our high risk sites. But of the sites that we sampled, in addition to our 6 additional sites, but of the sites that we sampled, we found Metasicaria across 12 sites, which are indicated for you there with the red stars. And then we also found 4 sites with Limnea tomentosa present. So with our snail surveys, we found 3 species of snails and an additional species of another type of mollusk. So the 3 snail species that we found were Limnea tomentosa, we found Glyptophysa species, and we found Physa acuta. This isn't atypical of what you'd traditionally find on a farm in New South Wales. Obviously, you're going to get some differences in prevalence to area, but this is pretty typical of what you'd find. And as I mentioned before, 4 of those sites had Limnea tomentosa. Unfortunately, all the Limnea tomentosa that we found were empty shells, so we weren't able to assess infection status. And we think that this is because we had drier than average seasonal conditions at the time of sampling because of the drought that we're experiencing. So when correlated against number of Metasicaria per kilogram of dry matter, we examined that against our soil pH, NDVI, soil conductivity, and elevation. So with that, soil pH, NDVI, and soil conductivity all came back positively correlated. Metasicaria was found over a pH range of 6.6 to 7.5. They were found between an NDVI of 0.35 and 0.5, and they were found over a soil conductivity of 20. So for during the sampling period, we, as I mentioned before, had drier than average conditions, but this actually probably proved favourable for our sampling as it created really clear distinctions between our dry and wet areas, as you can see in the picture here. And this would have really affected the differences in NDVI that we were seeing across the areas. So Metasicaria were found in a pH range of 6.6 to 7.5, and this is in line with the optimal range of pH reported, which is 6 to 7. However, while this is the optimal range, the broader range is a pH of 5 to 8. And we got a number of sampling locations that were below this 6.6, and the fact that we didn't find Metasicaria there is not indicative that this range is wrong, but more likely indicative that pH is obviously not the most determinant feature. So it's really well known and well published that NDVI is correlated with the amount of pasture biomass. And as we know, pasture is more likely to be more abundant where soil moisture is high. So it really makes sense that NDVI was highest where our Metasaccharide was also highest, and that we're going to have that correlation there. So where there were no Metasaccharide, soil conductivity is low, but yet Metasaccharide occurred over quite a wide range of soil conductivity. And this is likely because soil conductivity measurements change in response to quite a number of features in the environment. So this includes water holding capacity, cation exchange capacity, salinity and porosity of the soil. And these could all affect limnia, tomentosa distribution and Metasaccharide distribution differently. So from these preliminary results, it's clear that NDVI and pH are likely the most useful features for any future studies. As I mentioned before, we were a little bit limited in the number of sample sites and sampling that we had. So in any future studies, it's recommended that an increased number of samples per paddock are utilised. In addition to this, it'd be really good to test this for both within and between field variability, as well as testing during wetter seasons or on irrigated pastures. So machine learning modelling, once we have a larger data set, could be a really useful tool to analyse the type of data that we're collecting here. So not only does it increase the potential to expand modelling to different scales, but what machine learning modelling can allow you to do is identify the most determinant features of the environment and how much variation each feature is going to, each feature is accounting for. So when I talk about potential for expanding modelling to different scales, what I'm referring to is taking this from on-farm mapping to looking at regional risk mapping or national risk mapping. And so it's clear that the results from this study highlight the potential for NDVI to be incorporated into future forecasting systems. And this is a really valuable piece of information because, as I mentioned before, NDVI is so easily accessible. So this wouldn't be a Concepts in Parasitology seminar if I didn't take some time to talk about the course. So this course was extremely valuable for me. I'm fortunate enough to work with a lot of parasites in my job, but there's also limitations to what I'm exposed to. So the course, we had a great kind of distribution of research backgrounds with the participants in the course. We had the human side of parasitology, the wildlife side of parasitology, and the industry or the veterinary side of parasitology as well. So that was really valuable. I learned so much, and I can come to these conferences now, sit through like a real variety of presentations and understand a hell of a lot more than I would have before this course. And I think as Alex kind of alluded to before, the biggest thing that I took away was the time that these mentors and people in research took to come and teach us and mentor us and engage with us. But not only that, all the participants, I learned so much just from interacting with all the other participants in the course. It was such an encouraging learning environment, and everyone kind of stays in contact and really supports the growth, and we're all excited for each other's achievements. So I'd like to acknowledge the contributions of my co-authors, Derek Schneider, Dr. Lillian Makundiwa, and also my supervisors, Dr. Tommy Luong, Dr. Emma Doyle, and Dr. Les Gabor. I'd also like to thank all my colleagues that I've worked with over the years at Yarrandoo for their continued support. And I'd like to thank the Australian Society of Parasitology for inviting me to be here and be part of this showcase today. The Concepts in Parasitology course is not only a great development opportunity for early career researchers, but it's also a great example of the mentorship and the collaborative efforts within the world of parasitology. So thank you to Dr. Alex for all your hard work with the course, and I'm sure you'll do a great job, Stuart. Thank you.
Thank you very much, Tom. Any questions?
The effect of climate
You mentioned the effect of seasonality on the assessment of risk. Can you predict how climate change is going to affect the distribution of liver fluke, and will it bring new areas into risk and change the survey practices?
That's a really good question. So there's a number of papers that look at the change in climate change and the effects on Fasciola hepatica. It is going to be something that's difficult to predict. So one of the things that we might see is that we have that it could reduce. It could become obviously reduce the distribution, but also there's the risk that what we'll have is we'll have long periods of dry, so we won't have the infection, but then we'll have these short, big periods of wet, so we'll get these big spikes in infection. Another thing that we see in drier seasons is that cattle in particular get pushed into wetter areas, and so they're more likely to be exposed. So we might see a change in the prevalence in different species and different types of agriculture. One thing that we definitely do see is that infection increase with irrigation practices. So while seasonality is a big part, our management strategies also play a big part in our infection risk. And then with those two snail species that I mentioned before, we might see a greater distribution of those snail species because, as I mentioned, they're better adapted to those tropical climates. And so we might see that Limnaea tomentosa isn't our primary intermediate host in Australia. So but there's definitely ways that our seasonality can also be used to help aid us with practicing this. So I could have used some of those forecasting tools to go, okay, this season isn't going to be a good season to look for Limnaea tomentosa. But I think a lot of these practices can also be utilised without looking at seasonality. By looking at NDVI, it's almost attributing to that because NDVI is going to be lower, and therefore it's already going to be when it's a drier season, and so we'll have it's already kind of included in that analysis. I hope that answers your question.
Dominic, congratulations on a really heroic piece of work, and it's important in terms of help farmers identify hotspots on farms. Have you considered looking for environmental DNA, snail and fluke environmental DNA? Because as you know, there's now quantitative PCR tests for detecting DNA. That might add value to your work.
Yeah, I definitely have. That was actually my original strategy that I was going to look at. But unfortunately for me, I have to kind of consider how I fit my PhD into my full-time job. And so for me, looking at Metacicaria in the pasture was easy for me because I could kind of chip along at that maybe like a couple of hours a day at work, or on my PhD, I kind of do a day a week generally. And I could do that on that day with the facilities that we have available. But we've also talked about potentially collaborating with someone who might want to do that aspect of the project for any future trials that we do. But yeah, definitely for this validation work, be a very useful tool.
In the interest of time, one more.
Yeah, so I had a question. So the populations of intermediate hosts seem to sporadically, they seem to come out of the blue and then disappear. Mm-hmm. So how long do the Metacicaria persist in the environment after a population is gone?
Yeah, so that really is dependent on the environmental conditions. So the longer that they're maintained in a moist condition, the longer they're going to be there. And the temperature is a big factor, but generally they can last in the environment. It's generally accepted that about 6 months, but it can be longer or shorter dependent on the temperature or moisture conditions.
Thank you very much, Dom.
Thank you, Alex.
Okay, next person. So I have forgotten. Dom attended the course in 2016. Next person is Adelaide Dennis, who was in the first course in 2014. She's currently studying medicine at the Uni Melbourne, and that more or less sums Adelaide up. Adelaide has so many interests, and so she's interested in teaching, in research, in medicine, biology. So before she did her PhD at ANU and in biology, she loves science, but she also always wanted to do medicine. And during her undergrad studies, she fell in love with parasites. And so her big dream is becoming a clinician and a scientist. And she said to me that if anyone here in the audience manages to do both, she would like to speak to this person. As in terms of favorite parasite, that's hard for her to choose, but since she's most familiar with Plasmodium, that's probably her pick. And outside, as you can see, it's ultimate frisbee. So Adelaide, please come up on the stage, and I'm looking forward to a good talk.
Do you reckon you could help me bring up my talk? Alrighty, so I will be telling you a little bit about some of the highlights from my PhD thesis, which was on PF-ATP4, a transporter in Plasmodium, and the biochemical signature of compounds which seem to inhibit this transporter. But first, I'd like to give a little plug to the Concepts in Parasitology course. I did this course in its first year back in 2014, and it made a great impression on me. I think Dominic really summed up the course, and it just really represents all the facets of the ASP. I met vets and people in industry, government, incredible academics, and wonderful peers, which I see every year at the ASP conference. As a city slick and lab rat, I was absolutely mortified to be used as live bait for ticks, but I was talking to Amanda and she was like, "That's totally fine." And I was like, "Yeah, it's fine." But one of the really cool things we did was just go down to the beach and grab some sea snails. And I was mortified to learn that sea snails are full of parasites. And I don't know if you can see them very well here, but they're also on this thingy, this banner. But yeah, it was really cool just jumping under the light microscope, seeing thousands and thousands of Metacercaria of whatever species they are. Please let me know, because I actually don't know. And I was very thankful that we didn't have to count these ones. So there were a lot of them. I work on malaria. Multi-drug resistance is a problem in malaria. Now in Cambodia, which is shown in red on this map, we have parasites that are resistant to 4 of the different artemisinin-based combination therapies. So this is really alarming. In 2016, there were nearly 500,000 deaths and over 200 million new cases of malaria. And this is devastating. In response to this emerging multi-drug resistance, the kind of global community got together and started doing these humongous high throughput screens of hundreds of thousands of compounds in a kind of desperate effort to find new anti-malarial compounds. And some of these screens were done on the asexual stage of the parasite's life cycle, and basically hitting the parasites with each of these compounds and trying to find out which of these compounds led to parasite death. One of the compounds that did end up killing the parasites very effectively was this spiroindolone called KAE609, and you're going to be seeing a lot of this compound in the rest of my talk. So in 2010, Rotman and colleagues published a paper on the spiroindolones. It was all very exciting. The spiroindolones are very good at killing parasites. And to find out what the target of these compounds was, what Rotman and colleagues did was expose the asexual stage parasites to KAE609 at sublethal doses. This killed off a bunch of the parasites, killed them off, and then eventually we have a parasite which is resistant to the compound. You grow up these parasites, you sequence them, and what they found was that these parasites harbor a mutation in the transporter PF-ATP4. So in 2010, there was a great buzz at the ANU because Natalie Spillman, who was working in Kieran Koenig's lab at the time, was actually already working on ATP4. And I was in second year undergrad at this time, and Kieran kind of put aside half of his biochemistry lecture to basically tell us all about this thing, which was very exciting and probably how I ended up in his lab in the end. But Natalie went on to find that the spirulina indolents do seem to target ATP4, and she characterized this really beautifully in this 2013 paper. And I'll tell you a little bit about that. So Natalie showed that ATP4 maintains the low intracellular sodium concentration inside parasites. So here we have an uninfected red blood cell, and it likes to maintain a very low sodium concentration in the face of high sodium outside. And this is kind of the situation that's happening in our bloodstream. This unfortunate unfortunate red blood cell becomes parasitized by Plasmodium. And as the parasite matures, it induces the formation of these new permeability pathways in the erythrocyte membrane. And these new permeability pathways allow the movement of sodium down its concentration gradient and into the erythrocyte cytosol. So suddenly, the parasite, which itself wants to keep its sodium very low, is doing so in a high sodium environment. And we think that it keeps its sodium very low using PF-ATP4. So sodium is constantly leaking down its concentration gradient through into the parasite via unknown pathways. And then PF-ATP4 uses the energy from ATP hydrolysis to pump this sodium back out again. You'll also see that it imports protons at the same time. When we add KAE609, the spiroindolone, which we think inhibits ATP4, what you see is that the sodium rapidly increases inside the parasite. And that's because the sodium is rushing down its concentration gradient into the cell, into the parasite, but there's no way for it to get back out again. And so the cell fills up with sodium. And from some work, this is work that was originally done by Natalie, and this is how we measure the sodium inside the parasites. So we've got our asexual stage trophozoite stage parasites. We're going to isolate them from their red blood cell membrane and load them with a sodium-sensitive dye. The blue squiggle that you can see in the bottom left-hand side of the graph shows the resting sodium concentration in this population of cells, which was at roughly 5 millimolar of sodium. We add the vehicle control DMSO and we see that there's no change in the sodium concentration. But when we add KAE609, we see this immediate increase in sodium. So that's pretty cool. I mentioned before that ATP4 also imports protons. So now we're going to have a look at what the spiroindolines do to the parasite pH. So once again, we have our trophs. We're going to isolate them from their red cell host and load them with a pH-sensitive dye. These parasites are sitting at a pH of about 7.4. That's their resting pH. That's the blue squiggle there. And then when we add KAE609 to block ATP4, we see this immediate alkalinization. So after the original paper on the spiroindolones was published in 2010, there was kind of like an explosion of papers, all having new compounds which seem to inhibit ATP4. So we had the pyrazolamides come out in 2014, followed shortly after by the dihydroisoquinolones. Also in 2014, Adele Lehane, who now runs her own lab at the ANU, found many new ATP4 inhibitors in the malaria box. And then we had the aminopyrazoles. And this really solidly positioned ATP4 in the category of novel drug target. So I started my PhD back in 2013, and I'd like to share with you a few of the highlights from that journey. The first is a phylogenetic analysis that I did of PF-ATP4 and some of its relatives. I'd like to tell you about the role that cell swelling plays in the mechanism of action of PF-ATP4 inhibitors. And finally, I'd like to tell you the results of a screen of the pathogen box that I did looking for yet more ATP4 inhibitors. So firstly, the phylogenetic analysis. This was really prompted by a paper. This is the first paper kind of characterizing ATP4, and this was published in the year 2000. And firstly, you'll notice that it was characterized as a calcium ATPase, and I basically just told you it's a sodium ATPase, so that was a bit different. And it also says that ATP4 belongs to its own unique subclass that's unique to apicomplexan organisms. In this phylogenetic tree, ATP4 was kind of all by itself, and there weren't very many proteins in the analysis, so I kind of worked to expand this a little. So ATP4 is a type 2 p-type ATPase. We can further characterize these type 2 p-type ATPases into their subgroups A, B, C, and D, and these have been well characterized previously. Subgroup A and B are both calcium ATPases. So if we think ATP4 is a calcium ATPase, maybe it'll group with those ones. And then subgroup C and D include some sodium transporters, including subgroup D, which is an interesting one. It's basically sodium. It includes sodium ATPases that work in a similar way to ATP4. So we're looking carefully at that. What you can see in this garish background is the alignment of the amino acids used in this study. I basically took the amino acid sequence from ATP4 and many of its closest homologs, as well as the kind of representative proteins in each of these groups from model organisms. And I created a phylogenetic tree. And in this tree, we can see subgroups A, B, C, and D pop out. But what happens is that ATP4 consistently pops out in its very own subgroup, which I have creatively named ATP4 type ATPases. So this subgroup contains the Plasmodium ATP4, some close relatives in the apicomplexan group, as well as some representatives from the chromerids and the dinoflagellates. So it really represents a mesozoan kind of specific subgroup of type 2 p-type ATPases, that was a pretty cool find in my PhD. I'd like to go on now to tell you a little bit about the work I did on the role of cell swelling in the mechanism of action of ATP4 inhibitors. So we know that when we take our trophozoites, we isolate them from their red cell, we load them with sodium-sensitive dye, we check on Ka609, we know that they fill with sodium. And from our principles of osmosis, you think maybe there's some water being drawn into these cells, and that would lead to a volume increase of these cells. So we bought a Coulter counter, which is cool. This instrument here, it costs about 10 times more than my car, which is crazy because it's really simple. Basically, this is a beaker filled with cells and a saline. Within this beaker are 2 electrodes. One of the electrodes is surrounded by a glass tube with a tiny aperture in it called the aperture tube, and it's vacuumed. And so we can draw up 1 cell at a time through this aperture. And the electrical impedance created by the movement of these cells through the aperture is directly proportional to the cell volume. And you get a graph that looks like this. So I'm really looking at populations of cells in all of these experiments. We can fit this with a Gaussian distribution, find the median, and then we've got our population cell volume. And I used this method a huge amount during my PhD. So the first thing I want to tell you about is the effect of KAE609 on isolated trophozoites. So these are the ones that don't have their red cell host anymore. Here we're looking at the volume of the cells over about 25 minutes. And in the presence of the vehicle control, there's no change of volume over this time. But when we add KAE609 to block ATP4, we see that their volume increases. And for the isolated trophs, this kind of plateaus after about 25 minutes. We think that this increase is happening because of the sodium. And so what I did next was to test whether sodium was required for this cell swelling. And just to show this data in another way, here we have the percentage volume change after 15 minutes. The DMSO control caused no change in volume, whereas the KAE609 exposed cells did increase in volume. Here's just some controls with artemisinin and chloroquine. So we know that cells aren't swelling just because they're dying, which is an effect of cells dying. We know that this is specific to the KAE609. And then when we suspend the cells in a sodium-free medium, we find that it completely crushes the swelling effect. So we have no swelling in the absence of sodium. So we can conclude that KAE609 causes the cells to swell in a sodium-dependent manner. So that's pretty cool, but cells obviously don't float around in your bloodstream without their erythrocyte host. So here we have the cells. I'm looking at their volume this time, but I'm looking at the entire infected erythrocyte. So here we have their volume. In the presence of DMSO, they're sitting at about 90 femtoliters, and they don't really change over 60 minutes. But when we add KAE609, we see this increase in volume again. And in this experiment, it hasn't really plateaued after 60 minutes. And you think, okay, cells are swelling. What happens when cells swell? Well, they start to enlarge, and maybe they're going to be more fragile, more likely to burst. And to test this in an in vitro fashion, what we did was a lysis assay. So I took these intact infected erythrocytes and I suspended them in salines of varying tonicity from 0 to 1, where 1 is 300 milliosmolar. So that's just like a really happy saline. The cells should be great in that. And as you reduce the relative tonicity, you're going to start seeing cell lysis, which here I just measured looking at for the absorbance of hemoglobin that's released when these cells lyse. And so as we reduce the tonicity, the cells start lysing. And when we do the same thing for cells that are exposed to KAE609 for 1 hour prior to doing this experiment, you can see that the relative tonicity required to burst 50% of these cells actually increases slightly. So these cells have become more osmotically fragile. So we know that the cells are swelling and they're becoming slightly more osmotically fragile. In the in vitro assay, I wanted to find out whether this increase in cell volume had anything to do with the mechanism of action of KAE609. So what I did was kind of play around with the osmolarity of the growth medium in which these cells were placed. So this kind of regular medium is at 293 milliosmolar, and by definition, these cells are at 100% volume. When we add KAE609, we see about a 10% increase in volume, and that's shown in this data here. I then put the cells in a higher osmolarity medium, and these cells were then starting smaller, so they're at about 90% of their normal volume. And then when we add KAE609, they're not swelling as much. So in this case, I wanted them to swell back to kind of their normal volume. And that's shown in this data here. You can see that the cells start smaller and they swell, but they're not swelling as much. So I performed some growth assays in these two media. The first growth assay I did was in the normal media. You can see percentage growth over increasing concentrations of KAE609. And for the cells in the normal medium, we saw that it took about 0.6 nanomolar of KAE609 to kill 50% of the cells. I then did the same assay in the high osmolarity media. So remembering that these cells start smaller and they don't swell as much. And in this medium, I saw a small but consistent increase in the IC50 to KA609. So now it required about 0.9 nanomolar of KA609 to kill 50% of the cells. So why would cell swelling kill cells? The obvious thing is that, maybe the cells are bursting. I tried really hard to find cell lysis in vitro and I couldn't. But that doesn't mean that they're not bursting in vivo because of the perhaps increased osmotic fragility and the shearing forces of being in vivo. There might be some kind of consequence for splenic clearance. But really fundamentally, if you increase something with water, you're essentially reducing the concentration of every other solute. So that could have wide-ranging effects.